In aerobic respiration, we're going to take one molecule of glucose and break all of the covalent bonds that are holding the carbons together in the glucose. And with the help of some oxygen as our final electron acceptor, we're going to end up making 6 carbon dioxides and also some water. The energy that's released when we break all of the covalent bonds holding the carbons together in the glucose, we're going to take that energy that's released and use that to make some ATP to temporarily store that energy. When we break the bonds of the glucose, we're not going to break all the bonds at once. So we're going to do this process over 4 steps. So we've got glycolysis, which will be our first step, and then we're going to form acetyl-CoA, Krebs cycle, and then our last step will be oxidative phosphorylation. The first step of aerobic respiration is glycolysis, and if you look at the name glycolysis, we're going to be splitting glucose in half because glyco is referring to glucose and lysis means to split or break apart. So what we're going to do is we're going to split glucose in half into 2 pyruvates. So our 6-carbon glucose, we split into 2 pyruvates, which each have 3 carbons. So that's glycolysis. There's 3 different glycolysis pathways that you should know the names of. The Embden-Meyerhoff-Parnas pathway, that's the glycolysis pathway that you've learned before. So we just give it a different name in microbiology, but it's the same one you already know. The Entner-Doudoroff pathway is a pathway you don't know. So this one is used by some prokaryotes. And then the pentose phosphate pathway is a new one you haven't learned before either. And this one is used by both prokaryotes and eukaryotes. So let's start with the Embden-Meyerhoff-Parnas pathway. So this is the glycolysis pathway you already know. Very important to note that this does not require oxygen. So glycolysis does not require oxygen. So when we get to fermentation, remember fermentation is a way to make ATP when there's no oxygen available, and basically all we're doing in fermentation is we're just doing glycolysis. So that's important to know. You do need to know where glycolysis is taking place in prokaryotes and eukaryotes, and in both of them, it's occurring in the cytoplasm. And we're going to start with 1 molecule of glucose, which contains 6 carbons. That's what the 6C stands for. And we're just going to split the glucose in half into 2 molecules of pyruvate. And each So each pyruvate molecule will have 3 carbons each. And this is an exergonic, meaning that it's going to release energy, and catabolic reaction, meaning that we're going to take a larger molecule and break it into smaller pieces. The energy that's going to be produced, which you should know, overall we're going to have 2 ATP that we're going to be producing by substrate level phosphorylation. Remember, that just means you've got an enzyme that's going to take a substrate and take the phosphate group off the substrate and put on an ADP to make ATP. And then we're also going to be making 2 NADHs. Remember, NADHs are electron carrier. And those electrons are being ripped off glucose, and then we put them onto our electron carrier. And then NADH will then later on drop those electrons off at the electron transport chain in our last step of aerobic respiration in oxidative phosphorylation. And then they'll release energy there, and we end up making lots of ATP in that last step. The first phase of glycolysis is the energy investment phase. So you do have to put 2 ATP in to start breaking the glucose in half. So you got to use some energy to do that. And then the second phase of glycolysis, you're going to be capturing energy. So you're going to get the energy back. So you do end up making 4 ATP by the substrate level phosphorylation, but you did end up using 2 ATP to start. So your net gain of ATP is actually going to be the 4 minus the 2 ATP, so you end up with a net gain of 2 ATP. And then we've got the 2 NADH that we produce by oxidating our glucose, so we're going to rip electrons off glucose. And then the energy, the electrons that are in NADH, they will be transferred to the electron transport chain, and our last step of aerobic respiration in that oxidative phosphorylation step. And then this is just a picture just showing the energy investment phase of glycolysis. So you can see that we're using up 2 ATP to start breaking the glucose in half. And then the energy capture phase of glycolysis, you can see that we end up making 4 ATP. And then so our net gain would be the 4 ATP minus the 2 ATP that we use. So we end up with a net gain of 2 ATP. And then we also have our 2 NADHs that we also made. And we end up making our 2 pyruvate at the end. Another glycolysis pathway is called the Ettner-Dowdoroff pathway, just named after some scientists that discovered this. All you need to know about this is if you see that name, you just know that it's another glycolysis pathway. So it's just another way to break down the glucose. This is just used by some of the prokaryotes. Eukaryotes don't use this at all. And instead of breaking the glucose into 2 pyruvates, we end up breaking the glucose into 1 pyruvate and 1 G3P. So we just abbreviate that G3P. And then the microorganism, the bacteria, could take the G3P and then convert it into pyruvate later on. And you don't have to memorize how much energy is being made this way. But notice that we do make a different electron carrier here. We've got NADPH. There's a phosphate group on this electron carrier, and we're going to explain about that in just a minute. All right, and then our last glycolysis pathway would be the pentose phosphate pathway. So I just want you to recognize that if you see this name, that you know that it's a glycolysis pathway. And both prokaryotes and eukaryotes do this pathway, so everybody does this. And in fact, our cells are doing this pathway right now. How it got its name is that when you're breaking down the glucose, along the way, you do end up making some 5-carbon or pentose sugars. And so that's where the name came from. A lot of these sugars are going to be pulled out and used to make other things. So like the ribose can be pulled out to make part of the RNA nucleotides, because the RNA nucleotides contain ribose as their sugar. And the other thing that's made here would be NADPH. So that's an electron carrier that is a little bit different in where it's going to be taking its electrons than the NADH that we've talked about before. The electron carrier NADH is going to take its electrons to the electron transport chain. And this is going to be found in the last step of aerobic respiration and oxidative phosphorylation. Electrons go down the electron transport chain, they'll release their energy, and then that will be used to make lots of ATP in that last step of oxidative phosphorylation. And then we have the electron carrier NADPH. What's different here from NADH is that there's a phosphate group on the NADPH. And these electrons, instead of going to the electron transport chain, they're going to be used for anabolic reactions. Because when you're doing anabolic reactions and taking smaller molecules and making a larger molecule, a lot of times you need to add electrons to help make that larger molecule. So we need a source to supply the electrons from, and so that source would be the NADPH for these anabolic reactions. All right, step 2 of aerobic respiration would be forming acetyl-CoA. Where this is going to be taking place, it's going to be different. Prokaryotes, this is going to happen in the cytoplasm. But in eukaryotes, they have mitochondria. So they're going to form their acetyl-CoA in the mitochondrial matrix. And they're going to start with 2-pyruvate, which contains 3 carbons each. And we're going to end up with 2 acetyl-CoAs, which each have 2 carbons. We're also going to be forming 2 carbon dioxides as a waste product, which in us, we just exhale that. And then the energy that we're going to be producing would be 2 NADHs, our electron carrier. And then where the energy or electrons in the NADH will be transferred to will be to the electron transport chain, which will happen in the last step of aerobic respiration, oxidative phosphorylation. And we'll use the energy released by those electrons to make lots of ATP in that last step. And this is just showing you one of the pyruvates, and we would actually have 2 pyruvates doing this, but here's just one of them. And this would be a eukaryotic cell because we're showing the mitochondrion, and then inside the mitochondria with the mitochondrial matrix, so this is where the formation of acetyl-CoA would happen. And you can see the pyruvate, we start with 3 carbons, and what we're going to do is we're just going to chop off one of the carbons, and that'll form carbon dioxide, our waste product. And then we end up with acetyl-CoA, which only has 2 carbons. And then we're also going to oxidize the pyruvate. We're going to rip some electrons off and put those electrons onto our NADH. And that'll be our energy that's going to be produced. So we would have 2 of these going on, and this is just showing you one of them. All right, step 3 of aerobic respiration would be our Krebs cycle. It's also called the citric acid cycle or TCA cycle. Where it occurs in prokaryotes, it's going to be in the cytoplasm again because they don't have mitochondria. And then in eukaryotes, they do have mitochondria. So what happened in the mitochondrial matrix? And we're going to be starting with 2 acetyl-CoA molecules, which each have 2 carbons. And we're going to do 2 turns of the Krebs cycle. And we're going to end up with 4 carbon dioxides. So all of the carbons in our 2 acetyl-CoAs end up as the 4 carbon dioxides. And then the energy that we're going to produce would be 2 ATP. 6 NADHs and 2 FADH2s. And the FADH2 is just another electron carrier, just like the NADH. And then both of these will take their electrons to the electron transport chain in our last step, oxidative phosphorylation. Okay, this is just showing you the Krebs cycle. And you can see at the top, we're going to start with our acetyl-CoA. So this is just showing you one of the acetyl-CoAs because we can only send one of them through at a time, so we'd have to do this twice. So you can see the 2 carbons. So we'll attach it to a 4-carbon compound to make a 6-carbon compound. As we go around the cycle, notice we're going to regenerate this 4-carbon compound. So we're going to end up chopping off 2 of the carbons, and those end up as our carbon dioxide. And we're also going to rip electrons off and put them onto our electron carriers, NADH and FADH2. And we're also going to make some ATP along the way using that substrate level phosphorylation. Okay, and all I want you to get out of this picture is just to show you that with all of these intermediates, with the Krebs cycle, we can pull off a lot of these and the cell can make other things that they need. So maybe they need to make some nucleotides or some fatty acids or other amino acids. So there's a lot of things they can pull off the Krebs cycle and make other things that they need.